Yes, as many others have probably noticed, bioinformatics for biologists doesn't contain anything about machine learning, or very little about machine learning. So I thought which problem to introduce, which problem would be fit to introduce machine learning for biologists. And I came across my own favorite problem, protein sorting in eukaryotes. It's fairly simple to understand from a biological point of view. Everybody knows that proteins belong in different locations in the cell, and some even outside the cell. And there's a Nobel Prize in this field. Günther Blobel was awarded the Nobel Prize for the discovery that proteins have intrinsic signals that govern their transport and localization in the cell. Now, prediction of subcellular localization from sequence can be divided into basically 3 different kinds of approaches. You can use homology. Simply assume that your protein has the same localization as a homolog with known localization. Then you can try to identify the actual sorting signals, the zip codes of the proteins, and there are several examples of these: N-terminal, C-terminal, or internal. You can use global properties of the amino acid sequence. It has been observed that the amino acid composition of proteins is different in different compartments of the cell, and that fact can be used for a prediction method. And of course, there can be combined approaches. I will here concentrate on the sorting signal, the signal-based approaches, because they are the only way to be able to identify consequences of point mutations in the amino acid sequences. If you have a point mutation that alters the function of a sorting signal, it will probably not alter the homolog you find in a homology search, and it will probably not alter the global properties such as amino acid composition enough so that the prediction changes. And I will concentrate on 2 very well-known sorting signals. That is the secretory signal peptides and the transmembrane alpha helix. Now, what are they? Very briefly, the signal peptide is the entry ticket to the secretory pathway. It's probably the best known sorting signal of all. And it's cleaved off during translocation. So the bioinformatics question here is, given the sequence, is there a signal peptide? And if so, where will it be cleaved off? And the transmembrane helix is found in transmembrane alpha helix bundle proteins. And the bioinformatics question here is, given the sequence, do we have a transmembrane protein or not? And if it is a transmembrane protein, what is its topology? That is, which parts are outside and inside the cell and which are in the parts traversing the membrane. There are similarities between these 2 signals. We have here sequence logos showing the Shannon information of different positions in signal peptides on top and in transmembrane alpha helices on the bottom. And you can see that some stretch of hydrophobic amino acids is common to the 2 motifs. Now, how to predict those from sequence? Well, one approach is the artificial neural network, where a window in the sequence is converted into numbers using some representation, for example, sparse representation. And these are then sent through a hidden layer and to an output layer of computational units. Well, I should explain that it's not my intent in the proposed chapter to make biologists train their own neural networks, but to be super users of methods, so that they understand on a user level what is going on in a neural network. One example of a neural network method predicting the secretory signal peptides is signal P. It's composed of two different types of neural networks, one recognizing cleavage sites and another recognizing positions within the signal peptide, and they have then a combined score. And the newest signal P version 4.0 is from 2011, and it's I should report that it's still the best in distinguishing signal peptides from non-signal peptides. The hidden Markov model has some advantages when compared to neural networks. It has a simple grammar, and that is this grammar is ideal for solving the topology problem of alpha helix transmembrane proteins, because you can specify that an internal loop must be followed by a transmembrane helix, which must then be followed by an external loop. So you can't go from inside to membrane to inside again. You can build that into the grammar of your method. And it can model length distributions, so it can model the fact that transmembrane helices are typically longer than signal peptides. But it cannot, without adding some special features, learn higher order correlations between positions in your sequence motif. Now here an example is TM-HMM from 1998. There's this circular architecture modeling the helix core, the cytoplasmic and non-cytoplasmic caps and loops, and has models for the globular domains too. Now, TM-HMM is not the newest and not the best, but many newer TM prediction methods still rely on hidden Markov models or combinations of HMMs and neural networks. Now, what is important for the user of machine learning methods is to understand the problem of parameter estimation and overfitting. So if you have some points adhering to a model and you have some noise in your data, you can fit a 2-parameter model here. There's a good description, but the fit could be better. Now you can also fit a 7-parameter model, which gives better fit, but a poorer description and a poorer prediction ability for new examples. It's important to realize, also for the casual user, I will argue, that it's not interesting that a model can fit its observations, its training data exactly. So to function as a prediction method, it must be able to generalize, that is, produce sensible output on new data. And we can see that when training a neural network, performance goes up and up on the training set, and after a certain point, it starts going down on the test set. And this is very explicit in the process where you train a neural network, but it's not exclusive to neural networks. All statistical and machine learning methods can overfit. And when using biological sequences, the problem is even worse than that, because sequences are related in a tree-like fashion. And if the sequences in the test set are very closely related to those in the training set, we cannot measure a true generalization performance, because if these three were in the test set, then these two in the training set would be almost identical. So what has to be done here is a homology reduction, where you make sure that no neighbors defined as two closely related sequences remain in the data set, or alternatively, oops, I hit something wrong. Alternatively, you can use homology partitioning, keep all examples but cluster them so that no neighbors end up in the same set. So this is important to assess the trustworthiness of a machine learning method. Has there been a homology reduction or homology partitioning done when using the method? And that's important also for users to be able to read papers about machine learning. Now for the summary. The three classes of protein sorting prediction methods: homology, signals, and global properties. Signal peptides and transmembrane helices will then be used as examples of protein sorting signals. And neural networks and hidden Markov models will be used as examples of machine learning methods with some detailed descriptions of the methods signal P and TM-HMM. And finally, overfitting and homology reduction as a common theme for machine learning methods, also important for the casual user. Thank you.
May pretend that I'm a biologist and ask biology. So
yes.
I know that relatively few examples of signal peptides, maybe a few hundred, and half of them comes from AKL. So
no, there are several thousand.
Several thousand.
Yes, yes, yes. Yeah.
So the danger of overfitting, particularly if you go, let's say, to unusual GC content, genomes with unusual GC content, is it a concern? Or like, why should we assume that in every species, AXA would represent like standard cleavage site? Maybe there are species where it's completely different cleavage site.
Yes, that's absolutely true, and it's a particular problem for organisms like archaea, where we have on the order of 20 experimentally known examples. So existing prediction methods like signal P are certainly biased towards well-known organisms.
So, but is there today an experimental technique that would allow high throughput detection of signal peptides? Can we apply a technique and in a relatively cheap experiment and get 10,000 or 100,000 experimentally confirmed signal peptides? Because this technique is still, confirmation of signal peptides still remains a challenge.
It still remains a challenge, to the best of my knowledge.
I can see a formal biological definition of a signal peptide because it has a cleaved
Yes.
The version of the molecule and the initial. If you define a transmembrane segment, you actually go to the structure. You don't exactly know where position of the membrane is around the structure. The membrane is not very smooth, so you don't know. Actually, at the boundary, you don't know whether amino acids do belong to the segment or not. How do you deal with that? And what is the formal definition of the kinetic condition?
Yeah, that's so. Where do you define where a transmembrane segment starts and ends? And that's an unsolved problem, really. Different transmembrane topology predictors have taken different approaches to this, but it should be said that when comparing performances of topology predictions, people don't go very much into the details of whether they get the boundaries exactly right, but more whether they get the number of transmembrane helices and the orientation in the membrane right.
Okay.